Hello, this is Mr. Anderson, and today we're going to be discussing protein synthesis. So, what is DNA? Well, it's the basic building block of all organisms. It's the substance of inheritance, and it's made up of the four nucleotides A, T, C, G, which are adenine, thymine, cytosine, and guanine, respectively. It's a double helix structure discovered by Franklin, Watson, and Crick. And so, going with DNA, Chargaff's rule. It's by Erwin Chargaff, and it's a base pair ruling, meaning that there are equal percentages of adenine to thymine and guanine to cytosine. DNA is also a semi-conservative model. Each of the two daughter molecules will have an old strand and a new strand, and the old strand's from the parent, and the new strand's made entirely of new molecules. And so, DNA, RNA, and protein all follow the central dogma, which is the flow of genetic information in a cell, and it's how information moves from DNA to a protein. So DNA goes through transcription and also replication back into its own DNA, but when it goes through transcription, it becomes RNA, which then translates into a protein. So on RNA, there's four main types of RNA when dealing with protein synthesis. We have ribosomal RNA, or rRNA, which is the major component of ribosomes. We have transfer RNA, or tRNA, which carries the amino acids and has the anticodons to fit into the mRNA, which is messenger RNA, and it's a sequence of nucleotides that determines the sequence of the polypeptide, and it's made in the nucleus of the DNA during transcription. And then you have snRNA, or SNRPs, which forms the spliceosomes, which are used to cut out the introns from the pre-mRNA. So DNA replication follows three steps: initiation, elongation, and termination. So it starts it, stretches it out so it can replicate, and then terminates and binds back together. And so to translate or to transcribe from DNA to mRNA, it takes place in the nucleus, and a section of DNA is unzipped. RNA polymerase lays down nucleotides from the 5' to the 3', and the mRNA then leaves through the nuclear pores in the nucleus and enters the cytoplasm. And in transcription, you have your leading strand and your lagging strand, and your leading is the continuous transcription, which goes from the 3' to the 5' continuous. And then the lagging doesn't, and it forms the Okazaki fragments because it keeps having to go back and catch up with what it was doing. And then after transcription, we get to translation, which is in the cytoplasm and not in the nucleus. And so ribosomes attach to the mRNA, and they cover 3 codons at a time, and so initiation, the tRNA carrying an amino acid comes into the P site and bonds by base pairing its anticodon with the mRNA start codon. So you can see that bonding right here in this picture. And elongation, the second tRNA comes and binds into site A and bonds with the codon of mRNA. And then the 2 amino acids join with a peptide bond, and then you start growing a chain. And once it's all finished, connecting all the tRNA with the amino acids, it will terminate, and the ribosomes continue reading until it hits a stop, and then it will stop connecting amino acids together. And so the codons were the 3 bases, and they code for an amino acid. Some common codons are your start codon, which is AUG, and your stop codons can be either UGA, UAA, or UAG. The codons are complementary bonds with the tRNA anticodons, and there's less tRNA than codons, so the tRNA can bind with more than one codon. And there's the wobble hypothesis, which says that the anticodon with U in the 3rd position can bind to A or G, so you get a little discrepancy when you're in translation, I'm sorry. And so the ribosomes are the main site of protein synthesis, and they facilitate in coupling the tRNA with the mRNA. And the structure is RNA plus proteins, and there are 2 subunits, large and small. They have 3 binding sites in them. The A site, which holds the tRNA, the P site, which holds the tRNA with the polypeptide. The A site was holding the one adding to the chain, and then the E site, which is the exit site, which the empty tRNA leaves. And then building a polypeptide is the same 3 steps. The initiation, which is the mRNA, the ribosome subunits, and the initiator tRNA come together. The elongation, which is adding the amino acids that are based on codons together until they form a chain. And then the termination, which is the stop codon, or the releasing factor. So that's protein synthesis. This is an overview picture of all that happens. You have the DNA here, and it's unzipping and being transcribed into RNA that leaves the cytoplasm through the nuclear pores. And once it's out in the cytoplasm, the mRNA attaches to a ribosome, and it has the codons on it. And the tRNA comes into the ribosome, binds to the mRNA, and drops off its amino acid into the growing chain, and then it exits, and you get the amino acid chain, which is eventually turned into a protein. Thank you for watching. Hope that was helpful.